Hello students, welcome to EPG Pathshala. I am Dr. Savneet Kaur from Gautam Budh University, and today we are going to discuss about the module Cellular and Molecular Aspects of Stem Cells from the paper Molecular Therapeutics. Stem cells nowadays offer a great promise for the treatment of many end-stage diseases. They can compensate for the loss of damaged cells in various pathological conditions. But before knowing about the therapeutic uses of these stem cells, we should know the basic concepts about stem cells. So the major learning objectives of this module will be to define stem cells, what are stem cells, how they are derived, then what are different kinds of stem cells that are known, including totipotent stem cells, pluripotent stem cells, and adult stem cells or multipotent stem cells. Then the module will also introduce you to the concept of stem cell niche, which is the stem cell microenvironment, which regulates stem cell behavior in various manners. Now, first coming to the definition of stem cells, this is a population of undifferentiated clonogenic cells which are able to divide for an indefinite period. That means they are immortal and they have the capability to self-renew. That means they can produce cells of their own kind by the process of symmetric cell division. Besides producing cells of its own kind, these cells can also differentiate into different kinds of cells. That means they can produce more specialized, more differentiated cells by the process of asymmetric cell division. So coming to different types of stem cells, we have totipotent stem cells, pluripotent stem cells, and multipotent stem cells. When we say totipotent, these cells are formed immediately after an egg is fertilized in vivo. These cells have the potential to become any kind of cell, including the cells of the placenta. The placenta is a structure which nourishes and houses the embryo. So these cells can become the cells of the embryo as well as some extra embryonic cells too. When we say pluripotent cells, these cells come after a few divisions in the fertilized embryo, usually in the blastocyst stage, which is a stage which comprises of say around 50 to 100 cells in the embryo. These cells also can give rise to any cell of the embryo, but it cannot give rise to the extra embryonic cells. Finally, we have multipotent stem cells. These multipotent stem cells are present in the adult tissues and they usually cannot differentiate into all types, but yes, they can convert or transdifferentiate into the major few cell types, and that's why they are called as multipotent stem cells. So coming first to pluripotent stem cells. Historically, it was in the year 1981 when two independent groups, one from University of California and one from Cambridge University, they discovered about embryonic stem cells from mice, embryos, and these stem cells were shown to be pluripotent as they differentiated into all the adult differentiated cells of the mice. And after the discovery or the identification of the embryonic stem cells from mice, after 17 years, human embryonic stem cells were discovered, or human embryonic stem cell lines were developed by James Thomson, and in the same year, another pluripotent source of these cells was identified from the fetal gonadal tissues. Now, the embryonic pluripotent stem cells were derived from the blastocyst stage, from the inner cell mass, and the primordial germ cells, they were derived from the developing gonads in the fetus. These developing gonads, they house all the primordial germ cells, and the primordial germ cells, they give rise to the gametes in the adults. So they showed that the primordial germ cells behaved similar embryonic stem cells in terms of pluripotency and differentiated into almost all adult cells in the humans. So what are the criteria of pluripotent stem cells? They should be immortal. They should be propagated indefinitely in the pluripotent or the embryonic state. Now, these two features are also characteristics of the tumor cells, but unlike the tumor cells, the embryonic stem cells should maintain a normal
karyotype. That means there should be no chromosomal abnormality in embryonic stem cells. These cells should be able to form distinct colonies in culture, and they should be able to differentiate into cells of all three embryonic germ layers. That is, they should be able to form tissues from all the three germ layers, that is the ectoderm, mesoderm and the endoderm, both in vitro and in vivo. In vitro, this formation of cells is called as embryoid bodies, and in vivo, these growth of cells which contain differentiated cells of all three germ layers are called as teratomas. Now, besides the embryonic stem cells and the primordial germ cells, there are other pluripotent stem cells which can be isolated from the mouse embryo. But before we come to discussing these stem cells, I'll first tell you more about the blastocyst stage in the embryo. Now, the blastocyst stage in humans is formed almost after 5 to 7 days of fertilization, and it consists of say around 50 to 150 cells. And the blastocyst can be divided into the inner cell mass and the trophoblast. The trophoblast is the outer cell layer which will further form the part of the placenta. And the inner cell mass is the structure which gives rise to most of the embryo cells. Now, the inner cell mass further can be divided into two parts, epiblast and the hypoblast. The epiblast gives rise to the embryo proper, and the hypoblast, it gives rise to the extra embryonic yolk sac which provides nutrition to the developing embryo. Now, from the embryo, besides the embryonic stem cells, other pluripotent stem cells that have been developed are early primitive ectoderm-like cells, epiblast stem cells. FAB cells are nullipotent in vivo, but they can be converted to pluripotency in vitro by the treatment of FGF, activin and bio. So they are called as FAB stem cells. And besides these pluripotent stem cells, we can also isolate lineage restricted stem cells from the embryo, which are called as trophoblast stem cells and extra embryonic endoderm or the Zen cells. So all these cells can be isolated from the blastocyst stage of the mouse. And as the embryo development further proceeds to pre-gastrulation and gastrulation embryo, the number of stem cells that can be isolated decreases.
Now, first coming to human pluripotent stem cells or embryonic stem cells. Now, these cells, they closely resemble the mouse pluripotent stem cells called as the mouse epiblast stem cells. These stem cells are characterized by various parameters. We can characterize them by developmental potential. We can characterize them by using different transcriptional and epigenetic markers and also cell surface markers. When I say development potential, I mean that these cells should be able to differentiate into all three germ layers, cells of all three germ layers in vivo. This is called as teratoma formation. And in terms of transcription factors, we have certain core transcription factor which should be there in these kind of cells. These are OCT4, NANOG and SOX2. And then in terms of epigenetics, these cells are very different from the differentiated cells. The epigenetic by epigenetic changes, we mean the chromatin configuration, the histone and DNA modifications, which are usually in the open state or the active state in these cells as compared to the differentiated cells. And then in terms of cell surface markers, we have certain glycolipid proteins or antigens on these cells which make them different from the other stem cells or the other differentiated cells.
Now, the embryonic stem cells from mouse and humans, they are quite different in terms of their development potential, transcriptional profiles and cell surface markers. So this figure shows us basic differences between various pluripotent stem cells that can be isolated from mouse embryo and human embryonic stem cells. So as you can see in the figure, there are the transcription factors, 4 transcription factors, OCT4, NANOG, SOX2, KLF4, they are present in both mouse and human pluripotent stem cells. Then there's a difference between the human and mouse pluripotent stem cells in terms of the surface markers. The mouse stem cells usually they express the SSEA1 marker, which is state specific embryonic antigen 1. whereas the human embryonic stem cells, they express the SSEA3 or SSEA4 marker, which is state-specific embryonic antigen 3 and 4. And also human embryonic stem cells and some of the mouse pluripotent stem cells, they express a very high activity of alkaline phosphatase. And in terms of response to factors or growth factors in culture, the human embryonic stem cells, they show no response towards LIF, which is leukemia inhibitory factor, whereas the mouse embryonic stem cells, they show a good response to the LIF. We'll talk more about LIF in the next few slides. Instead, the human embryonic stem cells, they are very responsive to nodal and fibroblast growth factors. And then as in terms of development potential, as you can see, all the pluripotent stem cells, they are able to form teratomas or differentiated cells of all the 3 germ layers in vivo. Now, what are the factors which maintain the pluripotency of embryonic stem cells? Pluripotency means that these cells are maintained in a state of self-renewal by promotion or enhancing the proliferation of these cells and inhibiting the differentiation of these cells. So there are 2 factors, 2 mechanisms which control the pluripotency in these cells. One is the transcription factors and the other is the epigenetic factors. In transcription factors, as we discussed, the core set of transcription factors like LNOX, OX2 and OCT4, they will inhibit differentiation by inhibiting the genes which are involved in differentiation of these cells, and instead they will enhance the self-renewal of these cells by promoting the genes which increase the proliferation of pluripotent stem cells. And in terms of epigenetic markers, the epigenetic marks of the embryonic stem cells are quite different as compared to the differentiated stem cells. The epigenetic markers means the DNA chromatin structure or the histone structure, histone modifications. So usually histone acetylations and DNA demethylation, they are representative of an open or an active gene expression or chromatin state. So these marks, they signify or they characterize pluripotent stem cells and maintain these cells in a pluripotent state and inhibit differentiation. Now, it's equally important to maintain the pluripotency of these cells in culture conditions, and it is really a challenging task to maintain these cells in a pluripotent state in culture state. So initially these cells, the mouse embryonic stem cells, were cultured on inactive or irradiated feeder layers. Feeder layers are irradiated mouse fibroblast cells, so that the spontaneous differentiation of these cells, which is a natural phenomenon of embryonic stem cells when they are put in culture, the spontaneous differentiation of these cells should be avoided and they should remain in the pluripotent state. So initially the feeder layers, they provided some factors which kept these cells in the pluripotent state. But now, besides the feeder layer, we can culture these cells in presence of leukemia inhibitory factor, which is a cytokine which promotes the growth of these cells by inhibiting differentiation. Human embryonic stem cells, they can be cultured in the presence of human fibroblasts or human feeder layers. Even in the absence of human fibroblasts, we can culture human stem cells in the presence of some defined media components and ECM components. Now, in culture, these pluripotent embryonic stem cells, they can be coaxed or they can be differentiated into a plethora of different cell types, and thus they can be put into many applications in clinical medicine. Now coming to the multipotent stem cells.
After the embryonic stem cells, the next type of cells are the multipotent adult stem cells. Now these cells are undifferentiated, or better to say partly differentiated, and they are found in almost all postnatal tissues or the adult tissues. They usually remain quiescent or inactive for long periods, and they are activated only when they are required. Now these cells can undergo asymmetric division just like the embryonic stem cells, by which it can maintain its own population in the adult tissues, and at the same time it can differentiate into more specialized stem cells. So these have the potential to self-renew as well as to differentiate, but the only difference between pluripotent and multipotent stem cells is that these multipotent stem cells differentiate into cells of their own lineage. They are restricted to the cells of their own lineage and cannot go beyond borders.
The multipotent stem cells, they are present in almost all adult or postnatal tissues, and they help in the renewal and repair of the adult cells. They are involved in both physiological and pathophysiological conditions. Some of the examples include the bone marrow stem cells, the blood stem cells, brain stem cells, the epidermal stem cells in the skin, corneal and retinal stem cells in the eye, cardiac stem cells in the heart, and hepatic stem cells in the liver. So these multipotent stem cells, they are, although lineage restricted, that means they usually differentiate into their specific lineages which are present in the tissue in which they reside. But there are instances, there are studies which tell us that these cells can also transdifferentiate into other lineages if they are given proper specific conditions. And in the adult, the major functions of these cells are maintaining the normal cell populations and of course in helping the healing of the injured tissue. So they are involved in both repair and renewal of the adult tissues, and we can expand these adult stem cells ex vivo. They can be used for various therapeutic applications, like the bone marrow cells can be used as bone marrow transplants in the treatment of leukemias where your bone marrow has defective cells, or they can be used in the treatment of anemias where there are less of hematopoietic cells in the bone marrow, or like epidermal stem cells, they can be used for the treatment of skin burns. So first coming to bone marrow stem cells, there are many examples of multipotent stem cells, but here we are going to discuss about the bone marrow stem cells, which is the major reservoir of multipotent stem cells. We have three major populations, which includes hematopoietic stem cells, which give rise to all the blood cells through the process of hematopoiesis. We have mesenchymal stem cells in the bone marrow, which give rise to the support cells or the cells of the bone, cells of the cartilage like osteoblasts, chondrocytes, muscle cells, myocytes, fat cells like adipocytes. And then we have the bone marrow endothelial progenitor cells, which play a role in the regeneration of the endothelial lining of the blood vessels. These bone marrow stem cells, they are also present in the peripheral blood in small numbers, and they have also been isolated from the cord blood, where the cord blood has shown more numbers of these cells. So the bone marrow hematopoietic stem cells, they are present in very less numbers in the bone marrow. Almost one cell in 25,000 or 100,000 cells of the bone marrow represent hematopoietic stem cells. These cells are negative for lineage markers, lin negative. Lineage markers are actually a group of markers which are present on mature blood cells. So these cells do not show these mature markers, so we call them as lin negative cells. They are positive for markers like SCA1, c-KIT, which is a stem cell factor receptor, CD34. So these are positive for these three markers, and these cells can be divided into long-term and short-term HSCs. In culture, they have a rounded morphology just similar to lymphocytes. They are non-adherent and they are easily propagated in suspension culture. In the bone marrow, the hematopoietic hierarchy comprises of long-term HSCs giving rise to short-term HSCs, and further which give rise to the multipotent progenitors. The long-term, short-term and the multipotent progenitors, they form a part of the LSK compartment, which is lin negative, SCA positive and KIT positive compartment. And further, there are markers which can differentiate between these three cell types, that is the long-term, short-term and the MPPs. And the major important marker is the absence of CD34 on the long-term HSCs. So CD34 is the one of the major marker which is used for the isolation of these cells, but it is mostly the short-term HSCs which we get and culture in the lab as CD34 positive cells. So the MPPs, they further give rise to a common lymphoid progenitor and common myeloid progenitor. The lymphoid progenitor give rise to all the lymphocyte populations, and the myeloid progenitor, it gives rise to the granulocyte, and the erythroid progenitor gives rise to all the erythrocytes and platelet cells. After the hematopoietic stem cells, about 0.1% of the bone marrow cells, they are representing the mesenchymal stem cells, which express markers such as SCA1, CD31 and CD90. These cells are negative for hematopoietic and endothelial markers, so they can be easily differentiated from these two types of cells. Although these cells represent a very small fraction in the bone marrow, but the good part is that they can be easily isolated, they can be extensively expanded, and they can be easily differentiated into a number of cell types if they are given proper in vitro conditions. Now, these mesenchymal stem cells, they are not a unique feature of the bone marrow. Besides being present in the bone marrow and the blood fractions, they are also found in those tissues which have adult mesenchymal cells like the adipose tissue, lungs, amniotic fluid, placenta, even the dental pulp, tendons, skeletal muscles. So from all these sources, mesenchymal stem cells have been isolated and characterized. And one of the most attractive feature of these cells is their extraordinary immunosuppressive properties. So they are thought to be non-immunogenic because they do not express the major histocompatibility complex class 2 proteins and very less of MHC class 1 proteins. So this feature makes them an important candidate for cellular therapy in an allogenic setting. So you can easily transplant these cells in allogenic donors because they are very less chances of these cells getting rejected. The third major fraction of stem cells in the bone marrow are the endothelial progenitor cells. Now, these are progenitor cells because they are usually restricted to the lineage of endothelial cells. So, they reside in the bone marrow, peripheral blood and the cord blood, and they have been isolated from both mice and humans. They participate in endothelial lining regeneration in the blood vessels, and this process is called as neoangiogenesis, that is formation of new blood vessels, or they also participate in neovascularization or the formation of new vessels. They give rise to new vessels as part of pathological angiogenesis in tumor tissue, or they also give rise to new vessels as part of physiological angiogenesis, just like in case of myocardial injury when you have a major blood vessel obstructed and you need new vessels for blood supply. So these cells can be identified by surface markers including CD34, CD133 and VGFR2. And in culture, you can see the EPC colony looks like this, and it has a rounded interior, cells of round shape in the interior and the spindle shaped cell in the periphery. So this is a colony of EPCs after 8 to 10 days in culture. Coming to the stem cell niche, after discussing about the stem cell types, we should know how these stem cells are regulated in the in vivo environment. And the microenvironment of the stem cells is called as the stem cell niche.
Now let me introduce you to the concept of stem cell niche. Stem cell niche means the microenvironment in which the cells are housed. It refers not only to the anatomic location of the cell but also to the functional location of these cells. The niche components can comprise of different types of cells, different types of physiochemical environment like pH, ionic strength, or it can comprise of some secreted signaling proteins. So these proteins or these stem cell niche components, they regulate the stem cell behavior in various kinds, in both physiological as well as in pathophysiological conditions. In physiological condition, it supports the cell for self-renewal during normal cell turnover, and in pathophysiological or disease condition, it tells the stem cells to divide and renew for the damaged cell loss. So basically, the stem cell niche tells the cell when to divide and when not to divide. So that means it not only gives the signals for the stem cells to proliferate, but it also gives the signals to remain quiescent or inactive.
This stem cell niche refers to the anatomical and functional location of these stem cells, where interactions of the stem cells take place between the neighboring differentiated cells, The interactions of the stem cells take place between the extracellular matrix components, the growth factors, cytokines, and the interactions of these stem cells can also take place between the physiochemical ingredients like the pH, ionic strength, metabolites including ATP. And it is seen that both the stem cell and the niche component, they mutually regulate each other during development. So there are two kinds of signals which regulate the stem cells in the in vivo environment. One, they are called as extrinsic factors which form the part of the niche, and the other are called the intrinsic factors which include various transcription factors and the proteins that affect the epigenetic state of the stem cells. So both the extrinsic and the intrinsic factor, they together coordinate and regulate the stem cell behavior inside the body. The most well characterized of the stem cell niche is the hematopoietic stem cell niche. Now there are two niches which have been proposed for hematopoietic stem cells, the endosteal niche and the vascular niche. Endosteal niche is near the bone and the osteoblast cells, they form an important intrinsic factor, whereas the perivascular niche is near the endothelial cells. So the endothelial cells, they form the major cells which affect the hematopoietic stem cells in the vascular niche. Now it is seen that the endosteal niche, the hematopoietic stem cells which reside near the endosteal niche, they are more dormant, more inactive as compared to the stem cells which are there in the vascular niche. Actually, in the vascular niche, interactions between CXCR4, which is present on hematopoietic stem cells, and its ligand CXCL12, it increases the proliferation of HSCs, whereas this kind of interaction does not exist in the endosteal niche. In the endosteal niche, the osteoblasts secrete other signaling molecules like angiopoietin-1, Notch ligand, JAG1 and Wnt proteins. Activation of Notch signaling has been closely associated with the renewal of HSCs or the hematopoietic stem cells. Angiopoietin-1 signaling has been reported to be critical for sustaining the quiescence or the inactive state of HSCs in the bone marrow. And another protein, matrix protein, osteopontin, which is synthesized by osteoblast, is involved in negative regulation of the hematopoietic stem cells. And probably because of these negative factors, the endosteal niche is consisting of more inactive HSCs as compared to the vascular niche, as compared to the perivascular niche. Now, another key characteristic of the endosteal stem cell niche is the high extracellular calcium ion concentration, for which there is a calcium sensing receptor on the HSCs, and the high calcium ion concentration is also known to increase the proliferation of HSCs in the bone marrow. In terms of intrinsic factors, there are many transcription factors which are known to regulate HSC proliferation, which includes a homeobox B4 transcription factor, which is important in embryonic development, STAT5, STAT3, the transcription factors which are involved in the JAK-STAT pathway. And also we have polychrome group of proteins. One member is BMI1, which controls the gene expression through epigenetic mechanisms. This protein actually leads to the recruitment of histone deacetylases, leading to chromatin deacetylation, and it also causes histone methylation and of the DNA of the cell cycle arrest genes. The genes which are involved in cell cycle genes, they are inhibited by these proteins like the P16 and P53. So these proteins, they prevent the senescence of HSCs and maintain them in a self-renewal state by inactivating the cell cycle arrest genes. So these are the intrinsic factors which maintain the hematopoietic stem cells in a state of self-renewal or in a state of proliferation.
To conclude this session, I would like to summarize my talk. Stem cells are undifferentiated, non-specialized cells which can both self-renew as well as differentiate. Based on their differentiation potential, we can classify the stem cells as totipotent, pluripotent or multipotent. These pluripotent stem cells or embryonic stem cells, they are usually derived from the inner cell mass of the embryo at the blastocyst stage, and pluripotency is maintained by a set of various transcription and epigenetic mechanisms. Multipotent stem cells are present in the postnatal or the adult tissues, and they participate in the repair and renewal of adult cells. Major examples of adult stem cells or multipotent stem cells include the bone marrow stem cells, the blood stem cells, and the brain stem cells. In vivo, the stem cells are regulated by specific anatomic and functional locations, which is called as the stem cell niche or the stem cell microenvironment. And the stem cell niche components, they regulate the stem cell behavior by various cell-cell or cell protein interactions. And definitely the stem cells have immense potential for clinical applications. Thank you very much.